Thank you very much for sharing your ideas with us. Thank you. And let's hope we never find out if we are ready for another pandemic. Let's focus now our attention to vaccines against COVID-19 and the challenges we still never need to overcome. José Escribano is the founder and chief scientific officer of Alginex, Alternative Gene Expression, Spain, and is here to moderate this panel, which we are able to bring you thanks to the support of Bmw. He is joined by Mariano Esteban, chief researcher of the Pox Virus and Vaccines Group at the Spanish National Biotechnology Center, Jérôme Dallin, Senior Consultant of the European Medicines Agency Core Modalities Strategy Operationalization of Merck, Switzerland, and Arnaud Peggy, Scientific Affairs Director of Healthcare at Bmw, France. So we look forward to hearing all you have to say. Thank you very much.
Well, may I have the presentation, the short presentation?
El micro. Su micro.
Can you hear me? May I have the presentation in the screen? Thank you. Thank you very much. Well, good morning, everyone. Welcome to this panel, in which we will talk about one of the hottest topics of today, because the SARS-CoV-2 pandemic, which is the vaccines development. After the first session, we will have more discussions about this topic. I had the honor of coordinate this session, in which we will have three very interesting speakers that we will try to help you to understand how is the complexity of vaccine development. Not many people realize how difficult it is to develop and validate a vaccine and commercialize a vaccine. And then we will have, as I mentioned, three speakers. The first one, which is Dr. Mariano Esteban, which is a great expert in vaccines development, that will explain in detail how the viruses, how the vaccines develop a protective immune response. Basically, we have two options when we develop the vaccine, which is the first step, the decision about which is the technology to be used in the vaccine development. We have the vaccines divided mainly in two groups. One group is based on the vaccines that are produced ex vivo, and the antigens or the material is inoculated to the patient. And the second group of vaccines are those that develop the antigen in vivo, okay? We inoculate one material, infect the cells, or produce in the body cells the antigen to be recognized by the immune system. Mariano Esteban will explain in detail how the different options will develop nice immune responses. The second part of the discussion was related about the vaccine manufacturing. As everybody knows, one of the main issues in the present pandemic was the manufacturing of the different vaccine candidates that we have circulating today to attend all the demands in the market. To explain these issues and the options that we have, Dr. Jérôme Dallin from Merck will explain in about 10, 12 minutes some ideas or general ideas about the complexity of producing by conventional means the vaccines or using other technologies that are appearing in the last years that may substitute the conventional bioreactors, for example, the use of insects, tobacco plants, or microalgae in the production of antigens. And finally, one of the most important part of the vaccine development is the regulatory dossier preparation. There are a lot of issues related with the quality assurance, with the quality controls, with the clinical phases, etc. And Dr. Arnaud Paris from Merck will explain, we give an overview about the different issues related with this regulatory dossier. At the end of these presentations, we will open a general discussion between the people belonging to the panel, but also to the general audience, trying to get a nice discussion and giving ideas or discussion about these topics. Well,
Okay.
then starting with the first presentation, which is online, Dr. Mariano Esteban. Are you connected, Mariano?
Yeah, can you hear me?
Yes, we hear you. May we
Can
have
you see the slide?
may we have the faces of the people connected online in the screen, please?
Do you see my slide?
No, because it still is my presentation in the screen. Can you change
I'm
it?
sorry. Yeah, go ahead.
Please, may you change the presentation? Is anybody hearing me? In the control? Hello? Hello?
I can see your presentation, yeah.
Yes, that's the problem, that you don't eliminate this presentation, we cannot see you.
But you are making your own presentation.
Please, may you advise the people that is controlling the screen? No, this is the last. We need to see the people that is connected online. We had 2 speakers connected online. Please, may we have in the screen?
I'm checking if you
Now,
can hear
now.
me. Jerome speaking.
Now, we can see you now. Thank you very much. Okay, Mariano, the stage is yours. You may share the presentation and do it.
Can you hear me?
Yes.
Okay, perfect.
Oh,
Okay.
can you see the presentation?
Yes, we see.
Okay.
Thank you.
So, as you just pointed out, good morning and thank you very much for being attending this vaccine, COVID and beyond. So what I would like to highlight is basically how the immune system responds to this virus. And that it determines the success of a vaccine or not. The disease basically has 3 phases. Stage 1, which is the initial infection, followed by the pulmonary infection, and then hyperinflammation phase. So the key issue is that the virus should be stopped at the earliest stage so that it could not advance to develop this pulmonary phase or inflammation. And how we achieve that? And basically by the activation of the immune system. And the immune system has a key cells which control pathogens or any kind of insult to the host. And particularly the cells which take up the antigens or take up process the antigens, they can activate 2 types of cells. One are the T cells, which are in 2 classes, CD4 and CD8 T cells, and the CD4 cells activate, once they encounter the antigen-presenting cell, they activate another blood cells, which are the B cells, which in turn produce the antibodies, which are responsible for binding to the virus in circulation and and just stopping the progression of viral infection. But the key issue here is that to establish memory. The memory will determine how long the immune response will last and how able the cells are producing antibodies. And the other part, the other branch is the CD8 T cells, which they are also activated by antigen-presenting cells. And once they are activated, they can bind to the infected cells and produce holes and destroy. But also they establish memory. So these three types of cells, CD8 T cells, CD4, and B cells will determine whether an infection will succeed or not. And this is what the vaccines, they aim for. But basically, the important part is the nature of the antigen. In the case of coronavirus, we know quite well, we are familiar with this picture, and this is the protein which emerged outside of the viral particle. And this is the spike protein. This spike protein has a trimeric conformation, but this is the one that most important part that is included in the vaccines. So, and why is this protein important? It's basically because it's the key to enter the cells, and that it's done by binding to the receptor binding domain in the cells, which is another protein which is responsible for the recognition by this receptor binding domain of the viral spike protein interacting with this receptor. And that facilitates the virus penetration into cells. So what kinds of vaccines we have to activate these responses? So basically, now we have the Mrna Vaccine, which is a simple molecule, single-stranded molecule, and this is just the coding sequence for the spike protein, and that component is encapsulated into a lipid particle, which has several components, where inside you have the RNA, which is protected from the ribonucleases, which are quite abundant in an organism, in any parts of the system. So, and then we have another model that is being used, is the adenovirus, which is a DNA virus, and this is just the adenovirus particle, which inside has a DNA, and where we introduce the coding sequence for the spike protein. And also we have the pox virus, which is the one that we are using, and this was because these particles were used to eradicate one of the most lethal human diseases, which it was smallpox. What we do with all of this, basically, is just to try to present the antigen. If we look at as soon as we introduce by injection into a host the vaccine is going to produce the protein corresponding, which is, you can see in green, and this also is localized in the membrane of the cell, and this is going to trigger those branches of the immune system, both the B and T cell components. And that, if we see how long these vaccines last, in general, it's only a few days. This is one of the, if we inoculate intramuscularly in a mouse, so you can see in colors that the vaccine lasted for about 3 days and then disappears. So this is what happens with basically all vaccines, that it's just the vaccine is introduced into the host, produce the protein, and activate the immune system. What are the arms? How we measure this? Basically by ELISA, which is a measurement of the antibodies which are in the blood, and that we can see levels, and those levels dictate the strength of the vaccine. Basically, the higher levels, the vaccine is better. But also, we evaluated the neutralizing capacity of the antibodies, and this is just by binding the antibodies to the virus and see how effective it is in the neutralization. We can see here that it's basically knocked out the virus by the antibodies which are in the serum. But also we can evaluate those cells which I mentioned, CD4 and CD8 T cells, and then we can analyze memory, for example, that is quite effectively triggered in the case where we introduce a vaccine, any vaccine, that as stronger the response, the better will be the vaccine. And basically all of this, the final aim is to protect, and that we can see that in animal models, because we can do a challenge after the vaccine intramuscularly inoculation, and then we do a challenge after a month or so forth, and then we see how those animals are protected against the virus. In animals that are not vaccinated, the animals will die, but in those vaccinated will be fully protected. Okay, so eventually we come out with a series of vaccines, which are fantastic, and then where we can achieve efficacies above 90%, and this is incredible considering the vaccine. But what is the important criteria also? That the vaccines are also encounter the virus, and the virus tries to develop, introduce mutations which will confer some resistance to the vaccines or the immune system. And this is something that we are facing right now, and we are trying to see how the current vaccines or the future vaccines have to be produced in order to control our virus. So what are the considerations which I like to bring here? First, the durability, which is according to the efficacy of the immune responses for the different vaccines. How long will last these vaccines, or whether we need to immunize every 6 months? And this is something with a concern, because also we like the vaccines to be durable, and this is something we have to really improve. Also, to establish current vaccines can be effective against prevalent emerging variants of concerns, or need to generate viral-specific vaccines according to the VOC, according to how mutants emerge in the population, because we're vaccinating the whole world, and which means that more resistance will be emerging. And then to confirm if a combination of vaccines provide an immune benefit, whether we can combine one vaccine with another vaccine, and we know that in general, heterologous combination of vaccines is usually better in terms than homologous vaccination, but this is something we have to learn and we can discuss. And then what is this vaccination programs and these vaccines have provided is an incredible technology which really is beyond the control of the SARS-CoV-2, which also will affect other infectious diseases and non-infectious diseases like malaria, and also viruses like flu, HIV, and also cancer and other even degenerative diseases. So in all, we are facing an incredible progress of human beings and science as a whole, that we can now have technologies or approaches that can really face the different pathologies, not only viral, but also different diseases like cancer or other pathologies. So with that, I would like to finalize in time so that we can later on discuss. So thank you and for your attention.
Thank you very much, Mariano. Then we pass to the next topic that will be explained by Jérôme Dalin about the manufacturing of the vaccines. Jérôme, can you hear us?
Yeah, I can hear you.
Okay.
From far, but I can hear you.
May we have in the screen the faces of the online speakers? Hello, the control? Okay, thank you. Jérôme, when you want to tell us.
Yep. Can you hear me? Yes?
Yes.
Yep.
Okay, perfect. I'm showing my screen normally. So far, it doesn't work.
Here it is.
No,
No, that's mine.
that's mine.
No. Control,
Try
please.
to use your presentation, Arno, but I'm not sure I'm going to be ready.
Yes.
Okay.
Perfect. We move to presentation mode.
Yes. Well, here.
Is it working?
Yes.
Yes.
Thank you.
Okay, perfect. Let me hide this one. Okay, perfect. So thank you and good morning, everybody. So I'm Jérôme Dalin, and I'm part of Merck Group, and thank you for giving me this opportunity to speak today about vaccines. I'm just prepared a few slides to speak about vaccine manufacturing, and the first thing that I wanted to raise is the evolution of vaccine bioprocessing. I think we all know that Industry 4.0 was already really present before COVID, but with COVID we have seen a clear acceleration and a clear desire and need to move to concrete solution for an agile vaccine manufacturing. So finally now with COVID, plus the Industry 4.0, we have a new way of working. Let's focus first on the Industry 4.0, and let's go back in 1990s, where the industry and the facilities were made with very large floor space, very complex piping, very big skids, with an extensive preparation. And 20 years later, we started to see pre-designed facilities. That was already a good progress to reduce the floor plant, especially for non-productive activities. The clean rooms were smaller, and at the end, the infrastructure of the facility was much simpler. If we move to now, what we have right now is new facility generation, where we can make supply on demand. The majority of the rooms are now opened with a lower room classification. And thanks to that, it allows you also to have multi-product, especially with close processing. And with the progress that we made in biotechnology, we are also able to make smaller processes. Finally, what will be the future? That's the main question of today, post-COVID, which has been accelerated. So we will have ultra-compact and full equipment orchestration. And you have some pictures showing you which is already available or ongoing in terms of development. And we are going to have also modular and flexible facilities, where you have modules that you can add depending on what you do, if you need to increase your capacity, etc. And everything will be fully integrated, including process and QC. And an important topic is the close processing, so it will require full close process. Especially if you want to make like the ballroom concept, where you have a big open room, and if you want to make different processes into the room, you have mobile equipment that you can move, and if you want to make different campaigns, you will have to have everything closed. So just now focus on the COVID part. So we are facing a big outbreak, a big pandemic, and we face some challenges. The first one is time. So typically when you have to develop a vaccine from the beginning, from development to manufacturing, it takes years, sometimes more than 10 years. And finally, with the current situation, we have been able to make it much faster. An additional challenge is the cost. When you have to develop a vaccine, when you want to get the license, it costs a lot of money, sometimes more than $500 million. And at the end, you don't have any guarantee that you will get money back on long term, because there is a lot of competition in this market. And the last one is scalability and ability with a global pandemic to manufacture a huge amount of doses in a very short time. And capacity is really, really key in this specific pandemic. So what we have seen finally, we have been able to compress this time from 10 years to 1 to 3 years, which is really important. Technologies, innovations, as mentioned also before with my colleague, we are progressing, and we have specific technologies which really supported us during this pandemic. So single-use technologies, that's one of them. We also are able to improve the yield, and everything related to automatization of the process is really helpful too. And the last one, and we will come back to it just after this slide, that's the platform technology. Now we are working differently, and we have seen mRNA, viral vectors becoming more and more important in this pandemic, and we also see now VLP coming soon. So regarding the specific modalities, so the different templates. On the left, you have the traditional vaccines, which are historical, and we see now, let's say, a different approach, which is platform. So in the past, we were making one one vaccine for one drug. And now we have one platform which is able to make multiple vaccines. So that's a really different approach. I'm just going to use the laser to facilitate. So, and what we have in terms of platform, we have recombinant platforms, virus-like particle, conjugated vaccines, viral vectors for sure, and most recently, nucleic acid vaccines with RNA and pDNA. Something similar for all of them is the way to manufacture it. If you always have upstream to generate the antigen, the downstream to separate the antigen, and at the end, the formulation to combine everything together. You have maybe some differences for mRNA, which is a little bit different, but I will give you some details about that. So this is an example of a comparison between a classical template with inactivated process template, which could be flu, for instance, with mRNA platform. And the major difference between both is finally, at the end, you can see it's a biological manufacturing with the virus, which is not the case for mRNA. So we are making an enzymatic reaction. And this has a huge impact on many, many different aspects. On manufacturing time, on the regulation at the end, the way to produce, it's much easier to make it as an enzymatic reaction. The other steps, you can see that you always have upstream, downstream, and formulation, so make, purify, and formulate, but you have a big benefit with mRNA for sure. There is no doubt about it, and we all know that. On the other side, you have some challenges for sure. You know that the cold chain for mRNA is not easy to handle. You have to keep everything frozen, potentially with the current COVID vaccines. And also RNA-free products are also required because mRNA is really sensitive to RNAs. So just maybe to conclude, it was a quick presentation, but what finally is the main benefits of the traditional of the templates versus traditional vaccines? Sorry. So the first one is standardization. You always have the same template. You apply always the same process. So standardization is the most important. And for sure, it will have a direct impact on the development time that will be dramatically reduced. Also, the manufacturing changes will be significantly reduced as well. And at the end, that will ease you to get the final approval in terms of regulation when you will make your filing. So thank you for your attention, and I'm handing over to Arno.
Thank you very much, Jerome. Now is the turn of Arnaud.
Thank you.
That's going to explain us some points about the quality control and regulatory issues. May we have the last presentation, please, in the screen? Thank you.
Thank you very much. So first, thank you for giving me the opportunity to present at this very interesting conference. What I'm going to present here is, I would say, the last part of the first two presentations. It's really, I would say, the quality control and the release of vaccines. We've been talking about the development, the manufacturing, but it's very important also to consider the quality control and the release of the product. So very quickly to give you an overview of who is Bmw, because we are not a vaccine manufacturer, we are not process engineers, we are really dealing with public health overall, globally, with a clinical application and industrial application, and focusing really on three pillars, which are immunoassay, molecular biology, and microbiology, always looking for potential contamination, whether it's a patient or a product. And when we talk about product, we talk about the pharmaceutical product. So we've been in the industry for more than 25 years, and we are working with all the vaccine manufacturers for, I would say, all their quality control concerning potential contamination. Okay. Can you go to the next slide, please? I don't know why it doesn't work. Oh, okay. So I'm going to give an overview of where in the process of vaccine manufacturing there is a need for rapid control. because we've seen it in the COVID crisis. We need to manufacture a lot, manufacture fast and release fast products on the market and safe products. So this is really key for us to supply solutions that can make sure that you optimize your process, but that also give a high level of safety of your product for the patient. Next slide, please. So this is very quickly a typical manufacturing process for vaccines, bioproduction products, cell and gene therapies. There is always, like Jérôme was saying, upstream processing, downstream processing, and also the final release test, which is also very key. And during the whole process, it will be very important to implement what we call smart quality monitoring. So to make sure that right from the beginning, from the raw material that you use for any type of vaccines, up to the end, to the final fill and finish and the release of the product on the market, you implement clever control of your production process on product, but also on raw materials and process, and also on the operators that are going to do the manufacturing. And if you do it properly, then of course your product is going to be safe, and you are going to protect your process and your product as well. Next slide, please. So typically for the COVID-19 I would say crisis, there was a very specific constraint compared to traditional vaccines which take like 10 years to develop. For the COVID-19, we did not have this time, so it was very important to provide and produce a lot of vaccines and a lot of batches. Still to comply with all the regulatory approvals, with all the clinical trials, with all the data integrity. So it was really a challenge for the industry really to comply with all these criteria. Please. Thank you very much. Okay. Oh, thank you. Let's see. More. Okay, yeah, it works. Sorry, I need to go back now. And the volumes also to test was huge. Billions of doses were released and injected to different patients. So for us, as a solution provider for quality control and the process optimization, what was really important was really to develop rapid solutions. All the historical and traditional technologies are based on very old techniques, based on old microbiological processes. And we have developed specific technologies that are really fast and as good or even better than the traditional one to detect potential contamination. And this helped a lot to increase productivity, to reduce the risk of product failure and contamination, shorten also the QC processes. Some of the testing, like detection of final sterility test or mycoplasma in cell culture, could go up to 28 days, so it's huge in terms of quality control processes. We have developed solutions that can go up to, let's say, 1 or 2 hours to do those same tests. So that was really a key reduction in the process. And of course, the capacity was important to make sure that we could also, let's say, be aligned with the volumes that were produced for the market. So that's why it was important for us to develop those rapid solutions and to develop this approach of smart quality monitoring, not to do too much, but really to do the right quality control at the right place for the manufacturing. To increase, of course, the patient safety, to protect the product and the process of the manufacturing plant, to reduce the cost, clearly, also, because that was one of the constraints also for the pandemic, and to be able to give product on the market faster. That was also a key indicator for the production of vaccines. Okay, so it doesn't work very well.
Slide,
next slide, please. Sorry for all the issues. So now I'm going to give you a very rapid overview of different applications, different technologies that exist today to really optimize the process, the manufacturing processes, and the quality control of your product. And we call those rapid microbiology solutions because it's not based on traditional historical growth of bacteria, but it's really based on physical detection of contamination. Next slide, please. And the QC control and the QC evolution is also quite key. Like Jérôme was saying, the manufacturing evolved from old-fashioned manufacturing to Industry 4.0, and the QC evolved in the same way. So many years ago, it was purely manual quality control, and step by step, it went from manual to digital, and the future that we see is really automation and distribution of QC. By distribution, we mean quality control at the line, at the point of sampling during the process of manufacturing. That is going to be really the future. And this is really the quality control 4.0, like Industry 4.0, where we are developing solutions. And today, we are between the digital and the automation part. The next step will be really to have the almost 100% online and inline control of your manufacturing. Next slide, please. So here, a very rapid overview of different technologies that exist today that goes in this trend of digitalization and rapid technologies. Whether it's for the control of your environment or control of your process and product, there are different solutions available today that can really go much faster than the traditional techniques. And typically, for example, the mycoplasma solution with BioFire gets you a result in 1 hour instead of 28 days. So this is, I would say, the fastest technology that exists today on the field. And it's typically very important for all the vaccines that are based on cellular production. We have also another technology, Scanner DI, that takes a product and you control the sterility in 1 hour instead of 14 days. So you see that we are really reducing a lot this quality control time to help you optimize your process and also to improve the safety of your production. Because if you detect a contamination early on in your process, you don't have to go to the next step of manufacturing. You can stop and have a corrective action right from the beginning. So that's also increasing the quality assurance of your site. Next slide, please. Okay, so this is a kind of example where, in terms of value, this is important also to better control the processes. So I'm going to go to the next one. You will have the slides later. Next slide, please. And the optimal and I would say the final vision that we have, like I said, is really the QC 4.0, is really to create a contamination control strategy that integrates all the solutions all together in a kind of system that will be in the future able to predict contamination. So today we are really reactive. We are trying to detect if there is something in your product or in the process. The future is really looking at all the data that you control in your manufacturing process and defining a predictive model of potential contamination that could arise in your manufacturing process. This is not, I would say, science fiction. This is already existing for the food industry. So we are developing this for the food industry to redetect before the product is completely finalized if there is a potential contamination based on different parameters that can be controlled. And we are starting to develop this now for the pharmaceutical industry as well, because there is a lot of data that is captured and analyzed that we can integrate in the model to detect potential contamination. Next slide, I think. Yeah, so as a conclusion, so not only the development and manufacture of vaccine is important, but I would say the quality control, quality assurance is key as well. And microbiology is a key point and most often a bottleneck in the manufacturing and the release of the product. So we have developed a lot of innovative solutions to, I would say, to support the crisis, the pandemic with the COVID-19 vaccines that saw a huge acceleration of manufacturing and development. And of course, the trend is really to move to the Industry 4.0 and really to make everything more and more digital and to predict potential contamination that could arrive in your manufacturing. Thank you very much.
Thank you, Arno. Okay, we have still some minutes to open for questions and comments, not only for the participants in the panel, also for the general audience. We will have some microphones to facilitate the participation of the public. Well, I think we may start considering some points that for me are highly relevant. May we have the other participants in the screen, please? Hello? Thank you. Which is more relevant for all of you in terms of the selection of the platform to produce the vaccine? For example, is the duration of the immunity? Obviously, in the case of the SARS pandemic, the development speed was incredibly important, more than ever in this case. But in general terms, do you think that the duration of immunity, the safety of the platform that compared to the final product, the efficacy, when we are talking about a difference around 10% of efficacy or something like that, which is, in your opinion, the most important factor in order to select the right platform to produce a new vaccine?
If I may say, in terms of the current vaccines, we have really good vaccines that at present are able to control the disease and control virus infection. So this is very critical. And the fact that we reach levels above 90% for any vaccine, this is incredible. And I think that this is a big advantage. Now we are in the process of really trying to define the durability of those responses and whether we have to intervene by giving booster immunizations in the population, which is currently where we are. But what is important is to understand the mode of action of those vaccines, because we still don't know everything about how they activate the immune system and what consequences they do have in terms of the cell and signaling events or in the long run. So there are a lot of questions that we need to know. But what is very safe to say is that those vaccines are highly efficacious, so they are highly protected, and that is unique. The mRNA vaccines is a revolution in the field of vaccines, and we don't know how these vaccines will do it in the long run, but undoubtedly they are here to stay for a long time, and they are now going to be applied to multiple diseases. And I think this is a revolution in the vaccinology field.
Mm-hmm. And the reason because the mRNA vaccines are a revolution is related to the manufacturing process more than the efficacy that they have. Because what we see is that we may compare the immune response delivered by adenovirus vaccines with respect to the mRNA vaccines. And then probably the main difference between the two technologies is the manufacturing and the scalability, these kind of things. What do you think about?
Yeah, if I can quickly comment on that. So, as I mentioned, you have one enzymatic reaction versus a biologic to produce. So that has a clear impact, finally, on the way to manufacture on the facility itself. Biocontainment will be different. Equipment will be different. For the moment, the size is also different. So we are not talking about very large scale. I think the scale is much smaller. Maybe the bigger scale is to produce the plasmid DNA at the beginning of mRNA, but when you are just making the enzymatic reaction, that's pretty small. So that's all benefits that you have with mRNA, finally, if you compare to a classical vaccine.
Mm-hmm. Yeah. Okay, and if I may also, I would say on the process and quality control, the matrices are completely different. So the challenges are different from mRNA vaccine compared to typical traditional vaccines. So the solutions are not the same, and clearly we had to develop some specific application for that.
I see. And why we don't have protein-based vaccines against COVID? Because it's obvious that they will be a good candidate, but still there is not any in the market as far as I know. Do you think it's because the difficulty in producing the protein is because other platforms may deliver better immunity?
Well, in terms of the protein-based vaccines, I think that it's more related to the manufacturing process. And it depends on this, whether you are dealing with a full-length protein or whether you are dealing with the RBD or the S1 part of the protein. And I think this is the key issue. So how you are able to manufacture, and we see that there are some problems by some of the manufacturers, but I think that this is, I'm sure that we need to have the protein vaccine, undoubtedly. So, and that's why I say that we will see more the combination of vaccines. I'm sure that this is something that will happen because normally, when you are dealing with two different vaccines, whether it's an mRNA vaccine and adenovirus vaccine, for example, So we have seen that the combination is superior to is stronger, the immune response. And what you need to be effective, really, is how powerful you trigger the immune system and how durable it is. So in that case, an RNA, it acts in one way, but a live virus it acts in also in complementary ways. So it's broader, the immune response. So the combination will really, I'm sure that this will be perhaps implemented in different parts of the world.
Yeah.
Thank you.
And actually, we are working on the clinical side for the, I would say, to measure the efficacy of the vaccine on serological tests, specifically for those kind of projects to see the combination of vaccines, which one is going to give the best, I would say, protection and how long it's going to last. So it's not only, I would say, on the manufacturing, but on the clinical part, it's very important as well to develop those typical critical tests for a diagnostic test that will measure this response.
Exactly. Anybody in the audience want to participate and doing some comments or questions? Any of you, Mariano or Jerome, Arnaud, do you want to comment something?
Well, I think that coming back to the protein base, and I think there are companies which are producing rather shorter size molecules, and are just trying to organize in a way that those are better presented and they are more immunogenic. So it's something that we learn a lot. And just to put you back into, for example, the HIV field, where we've been working heavily, many groups, in terms to try to present the envelope protein in a way that can trigger a wider response and higher levels of antibodies, which could neutralize different clades. So this is something that I think the technology is here, but still we have to know better how to design the protein itself, and so which combined with the adjuvants, which are very few adjuvants, by the way, so but they are quite potent. They can really be used as vaccine. But I really think we need the protein-based vaccine for the SARS-CoV-2 and for many other diseases.
Thank you, Marino. Coming back to the technology platforms, what do you think is more important for the companies? Is the manufacturing cost, the simplicity of the process, the development speed, the downstream complexity? Obviously, the safety concerns are also important. What do you think that the companies look for, visualizing will be next in the vaccines development in the future in terms of platforms?
Yeah, I think cost potentially is really key, because you are making massive vaccination. So you have to produce a lot of doses in a very short time. So that being said, so the first focus is how to manufacture the maximum of doses in a shorter time and with a reasonable cost. And from the list that you mentioned, I think cost is driving a lot of discussion for manufacturers.
Mm-hmm. Today, I think there are some tools that are not adapted, like the current regulation sometimes doesn't apply in the COVID or in the pandemic time, because we've seen it with COVID-19, there was specific fast track for vaccines and for development of product and solutions. But the traditional regulatory process, I would say, or even the regulatory tests that are needed for product and for pharmaceutical product, they cannot apply every time because they are not adapted, and we need to adapt and to implement new technologies that are really more in line with what the new manufacturing process do, and we are not based on the, I would say, the historical traditional vaccine manufacturing, and it's really a question, like Jean was saying, of speed, capability, and I would say scalability, because you need to produce a huge amount of vaccines and at a cost that is quite reduced. So mixing the three of them, you need to find new solutions and new controls compared to what we are doing today.
And from the point of view of the regulatory agencies, do you think that they are more open now to accept new technologies in the vaccine production, or still they will be, whether we have a lot of concerns about the new platforms?
We hope they're going to accept more. No, they are moving. They've been moving at least already now for, it's not a vaccine, but for the CAR T cells or the cell and gene therapy. There have been a lot of evolution on those kind of products because the shelf life is very short. So there is no choice to have rapid solutions and rapid release of those products. So they have started with this, and now they are starting to consider applying those kind of same technologies to other products like vaccines, because it makes a lot of sense for the industry.
Okay.
And I think the authorities have been a little bit more flexible than usual, and it was mandatory if we wanted to stop this COVID pandemic. But what we expect is that when we will, and hopefully soon, come back to a normal situation, they will be less flexible. I think they learn from what happened, but potentially they will ask maybe for more specific things, especially if new platforms are coming. Just thinking about VLP, a virus-like particle platform. That's, let's say, the third one which is coming in this pandemic, and I think one manufacturer is quite close to reach the market. And then we'll see for the other modalities how it will be, but we expect them to be a little bit more, let's say, strict than this current situation.
But I think the more we will get those technologies applied to other diseases like HIV or cancer, it's going to be more and more accepted and I would say mainstream. So today mRNA is super new, but it's like biotechnology. Like 20 years ago, it was completely new, and now it's more and more considered as a standard manufacturing process. So I agree with Jérôme that they are going to maybe to come back to a more stringent overview of regulatory overview. But the more application, the more products are released, I would say the easier it's going to be.
Okay. We have a question.
It's working. Hello, I have a question related to more on the quality control of mRNA vaccines. Do you think that we are missing, or do we need to develop new tools, new device to assess the quality of this new mRNA?
This is probably the question is for
I
you.
don't see the person. I don't know where the person is in the audience.
Where is the person, please?
Oh, okay.
Okay.
It's a really good question. We know that the traditional methods apply to mRNA vaccines, but the whole concept around the speed and I would say fast release of the product need to have a development of specific solutions. So it's more, maybe not specific solutions for mRNA vaccines, but at least to validate the application and the compatibility of the product with the technologies that are already on the market. Today there is a lot of solutions available for, I would say, characterization, safety assessment of vaccines that can apply. And I would say the most important part is really to make sure that they apply and that they are compatible. If not, for sure, we will need to develop specific technologies for that.
Thank you.
We have another.
Questions? Yeah, two quick questions. One is, what should we expect from the new vaccines to really say that they are better to the ones that we have now? And the second is, now that most of the population in the developed countries are already vaccinating, what's your clinical plan for phase 3? Are you planning to go, to the less developed countries?
Do you want to answer this question?
To whom this is addressed, that question?
This is, well, your question was related about which are the plans if we are extending the application of the vaccines to developing countries, or we?
No, the second question.
I think that as in the industrialized countries, the vaccination programs, they really are reaching very high levels. And then, but in many other countries, like in Africa or other parts of the world, there is minimal population which are vaccinated. So it means that we have to really perform also trials with the new vaccines or the incoming vaccines in countries where the incidence of infection is high and the population vaccinated is very low. So that could accelerate, so you don't have to really go through these large phase 3 clinical trials, because we know which are important, the important correlates of protection. So as I pointed out, I think you need to really have a very good levels of antibodies and neutralizing capacity, and also activation of T-cell responses. So this is easily now evaluated in the vaccinated people. And that, I think that the regulatory agencies will be more prone to really not require this number of thousands of individuals to really go through this phase 3. So I think that now the phases are accelerated. So now phase 1, phase 2 are really just moving fast. And then just as you prove that the vaccine is safe and is immunogenic and triggers this neutralizing capacity, so that will really move further into the population. So I think that we will see more fast speed in the implementation of the incoming vaccines.
Okay, I
And if
think
I can add a comment
this is
regarding-
the last question.
Morning. I will have to do a question to Mr. Mariano Esteban. First of all, I want to congratulate you for your job, because you are doing one of the best investigations in Spain regarding to vaccines. So as a Spaniard, I am very proud of you. Thank you very much. And I also... I am the president of the committee of the Spanish National Award in Biotechnology, and you was a candidate last year, and I hope that you will win the next year. So that's for your own information. My question is regarding to the Spanish investigation in vaccines. All the Spanish newspapers, we are always reading that you are very close to get this vaccine. It will be the first Spanish vaccine. If you can tell us some information about the situation of your vaccine, I think from the CSIC, and you're working on the CNV, where you are working in the National Spanish Biotechnology Center. And the second question is that I read also in the newspapers, because I am also a member of the National Biotechnology News Association, that you have some problems with animals working with the vaccines. So please, if you can answer to this question, thank you. And again, as a Spanish, we are very proud of you.
Okay, so thank you very much. I highly appreciate your words. And for sure, we are trying to do our best. And we've been working on vaccines for many, many years, in particular with HIV. And this is highly difficult to get an effective vaccine, which we still are trying to get an effective vaccine, which we don't have it yet. But in terms of the Spanish vaccines, I think that this is an effort that has been done here in Spain, because we have very good groups working on different strategies, and I think this is a big advantage. We didn't have a pharmaceutical companies, largely because they were in the veterinary field, but unfortunately now several of those companies now are producing vaccines against SARS-CoV-2, which I think is very important for the country, because we have to have the manufacturing technology to really address any issue of an infection. And in terms of where we are now, so we have fulfilled the complete studies in terms of preclinical in the 3 animal models, so with very good results. We have published some results. We published the series of other papers in that. And for the vaccine, simply we just are responding to questions raised by the National Agency of Medicinal Agency, which is the Spanish, and this is, they ask a number of questions which we are responding to that. So this is as simple as that, and this is no problem with in terms of what the newspapers have just mentioned. But also there are already a Spanish company, which is Mrna, that has produced a protein-based vaccine, which is now they are doing clinical trials, and I think this is very, very, very good for the country. And then we will see other coming vaccines in the near future. So really, I thank you for your question and for your words.
Okay, we have to stop here. Thank you very much for the speakers, the participants in the panel, and also for the audience. Thank you very much, and enjoy this meeting. Thank you.
So thank you very much.
Now, thanks again to bringing us to date on the vaccines against COVID-19 and the challenge we face in that field. We'll be shortly holding the official welcome session. That will be in a few minutes, so see you then. It will be like in 5 minutes. So thank you, and see you then.